Perfectly audible and visible. Hi everybody. Okay. Hi everyone. How are you doing? Hi everyone. How are you doing? Hi everyone. How are you doing? Am I audible? Yes. Am I audible? Am I audible, everybody? Okay, so good afternoon. Welcome to the session, students. So today it is a bridge course for those who are preparing for NEET 2026. Okay, so trust me, you are going to get benefit from this session, because I will tell you that if, let's suppose, if you were not that sincere in class 11th, then how to compensate all the things in class 12th. This is what we are going to discuss today, and then we will talk about the basics of biology. Okay, so firstly I'll tell you that if, let's suppose, you were not you didn't study well in class 11th and you were not started up to mark in class 11, then how to compensate, how to manage that thing in class 12th. That's what we are going to discuss today. Okay, that is what we are going to discuss today. So first of all, welcome to the session. If you people are new to our channel, please do not forget to subscribe our channel. And I'm your biology educator Ambika Sharma, and let's start the class. Okay, so thank you so much for joining, everybody. Thank you so much for joining. Thank you, Afsal. Okay, so let's start the class, let's start the discussion. But yes, let me tell you, students, that from April 1st, your plus subscription will be for 9999, that is 10,000 rupees. And if you get it today, right, that is, today is the last day. If you get it today, right, that is on 31st of March, my dear students, then you can get it at rupees 4499. So if you are in class 12th and with your class 12th, right, you want to crack the NEET 2026, you don't want to take the drop, and you have decided that this year only you want to crack the NEET examination, right, and along with your board. So yes, we are here for all of you. The complete team of angels is there for all of you. So what you have to do, you have to join in our batch basically. So if you are in class 12th, for you people, we have the Nexus batch. But let me tell you here about the plus subscription as well. So students, when we talk about the Unacademy plus subscription, so basically what we have to do, let's suppose you enrolled in our batch. Okay, let's suppose you enrolled in our Nexus batch. Okay, right, because you want to prepare for class 12. But here in this batch, you are okay with the physics, you are okay with the chemistry, you want to study the biology from somebody else. But trust me, you're not going to do that if you will enroll yourself in my batches. Trust me on that. So let's suppose you want to read some other subject from some other teacher, so you will have the access of other batches as well. So basically, when you people enroll yourself in plus platform, it is not just the one batch that you people are going to get. You will have the access of all the batches in NEET UG category, right? You will have the access of all the batches that are running across NEET UG category. Trust me on that. So you just need to pay this amount once. Okay, so you will get the access of all the batches. You will get the access of all the teachers' notes in the PDF format, of course. Whatever content is available on the Unacademy platform, right, that will be accessible for all of you. Plus, at the end, you will get the access of our test series as well. So if you enroll in the plus platform, so let's suppose we'll start our batch, we'll finish your syllabus, right? Initially, we'll take 6 months to finish your class 12 syllabus. Then for the next, right, 4 months, there will be your class 11th revision as well. Okay, so in which batch you have to enroll? If you are in class 12th, you have to go for the Nexus batch. And trust me, right, even with the Nexus batch, students, even with the Nexus batch, whenever at the end we'll start our crash course, you will automatically get enrolled in that. So basically, you just need to buy the subscription just for once, and after that, whatever batches will start, new batches, old batches, you will get the right access of everything, even of the old batches as well. So that is the beauty of plus platform, and just for today only, you people will get it rupees 4499, and after that it will be at this price. So enroll yourself now, okay, without any delay. And our batch is starting from 4th April onwards. Do not forget about it. Okay, so if you want the hard copy of the content, you people can go for the iconic subscription. Here you can see and you can compare as well. Okay, so there is one more batch which is CBSE combo, right, where your CBSE board syllabus will be covered along with the NEET preparation. So you guys can see the price here. Okay, the link is there in the description box, and as I said, offer will end tonight. Okay, the offer will end tonight. Okay, so let's start, right? Let's start. Okay, so you tell me, how many of you think that you have justified your class 11 syllabus, and how many of you think that no ma'am, we have wasted it completely? We have wasted it completely. Tell me. Yes, please tell me. Yes, please tell me. No ma'am, wasted it. Akriti is saying I have wasted it. Ru is saying I have wasted it. Now what's your plan? You tell me. Let's interact and then we'll start the discussion. So Ruhi is saying wasted it, and yes, Amulo is saying wasted it. So basically, majority of students that are watching this lecture, they are saying that we have wasted the class 11. Now, what is motivating you to start again? Yes, what is motivating you people to start again? That we should start our preparation again. We can crack the NEET examination in 1 year. We can complete our class 11 syllabus and class 12 syllabus in this 1 year. What is motivating you? What is motivating you? Yes, please. Dream. Okay, my parents' dream. Okay, so basically it happens, right? Genuinely it happens. I repeat this line so many times that when we move from 10th to 11th right, a lot of things change, because in class 11th we have to do the self-study. We have to be self-disciplined. That is obvious, students. That is obvious. We have to do all that things. We have to focus on our studies, right? We have to be self-motivated, right? We have to utilize our time. That is obvious. That is something we have to do, right? That is literally something that we have to do. Okay, but many times what happens, bache, when students, they move from 10th to 11th, they are not, they basically they are not able to deal with that pressure. They feel like that it is too much, we will not be able to manage, and a lot of distractions are also there, right? Your social media, if you go to school, a lot of things happen and we get distracted. That is obvious. That is obvious, right? So in class 11, there are many students, right? There are many students who just, they just don't able to cope up. Okay, but it's fine. It's fine. Okay, fine. You have to. First thing that you have to understand today is that it's okay if you have wasted your 11th, right? You have to make peace with this fact. It's okay if I have wasted my class 11th. It's totally fine, because sometimes what happens that when we are in class 12th, then again we have to deal with the pressure. Now the boards are also there. We have to finish the class 11 syllabus as well. After 12th, the entrance examinations are also there. Again, the pressure is there. Okay, so sometimes what happens when we start our class 12th, we just think we will be able to manage, but what happened, bache, we are not able to again, right? We feel frustrated. We feel sad that why we have wasted our class 11. That it happens. It's natural. Trust me on that. It's natural. So the very first thing that you need to understand today is, and that is something you have to promise yourself today. Okay, literally, that is something that you have to promise yourself today, that it's okay, it's fine if I have wasted my class 11. Now I'm going to, right now I will start class 12, right? And that start should be a fresh start. No frustration, no regret, no tensions. First of all, tell me. So if you want me to continue this lecture, this is something that you have to promise me. Yes, this is something that you have to promise me that, okay, ma'am, fine. Whatever we have done, It's okay, it was past. Now we are going to start fresh, and we are not going to regret for whatever we have done in our past. Is that okay? Everybody aligned? If you are okay, just send me a thumbs up. Just send me a thumbs up in the chat section that yes ma'am, we are ready for that. Because if in class 12th also you'll keep thinking about class 11 stuff, then again you will be frustrated, and again you will just lose that thing. Okay, so the very first thing that you have to do is you have to make peace with the fact that okay fine, I was not able to study that much in class 11, but now I have to compensate that in class 12th. Okay, so that is actually going to be a fresh start, number one. Now in class 12th, what we have to do, how are we going to manage, right? How are we going to manage the class 12 syllabus? Because we don't know much. Yeah, we don't know very important concepts of class 11th as well. It's simple. See, your class 12, if I talk about the Unacademy platform, so your class 12th classes will start 4th April onwards. When will we start classes? We'll start classes 4th April onwards. Okay, 4th April onwards, I'm going to start the classes. Okay, in your Nexus batch. Now, what you have to do in that time? So you have to pick up the topics. Okay, because we have to follow a proper strategy. If you people will follow a proper strategy, then it will be easy for you guys to compensate. So first of all, in that particular time period, you still have 3 days or 4 days. In that 3 or 4 days, try to revise most important topics, right? What you have to revise? Most important topics of class 11th that are related to class 12th, that are related to class 12th. That is something you have to understand first. Okay, that is something you have to understand first. Trust me. Okay, so for an example, if I talk about biology, in biology, my dear students, right, do revise cell cycle and cell division. Okay, I'm coming up with a very short session series as well. You can follow that. Do revise biomolecules. If you can, go for plant kingdom, go for plant kingdom and morphology if possible for you, right? Revise these chapters before starting your class 12 syllabus, right? I'm giving you the examples of biology. Same for the physics, same for the chemistry. If you need something to understand class 12 physics is comparatively more interesting and easy if I compare it with class 11. It is as per my experience, okay? It is literally as per my experience. So this is what I want to tell you, my dear students, that you pick up the most important topics or the chapters of class 11th chemistry and physics that you need to understand before starting your class 12 syllabus. You can just put this thing on YouTube, you'll get multiple videos, right? You can check our channel, you'll get multiple videos for that. So this is what you have to do first. Okay, this is what you have to do first. Now, when we are in class 12, what we have to do? Let's say you have to start early. First of all, what you have to do? You have to start early because this time we have the boards as well. What do we have? We have the boards as well. So start in the month of April 1st. Start in the month of April 1st. Then, right, April, May, June, July, August, September. Trust me, for class 12 syllabus, especially for the biology, 6 months are sufficient enough. Still, let's say till October complete your class 12 syllabus. That should be your strategy. Okay, till October complete your class 12th syllabus. Then in the month of November start with class 11th, and do not start with the complete syllabus. Start with the most important, right? You have to start with the most important chapters. Okay, right? Most important chapters. So here in our Nexus batch, after completing your class 12 syllabus, we will have, right, we will have class 11th revision as well. We'll have class 11th revision classes as well. And meanwhile, you people can, right, revise these two chapters from your notes as well. This should be our strategy. Now in the month of December also you'll have exam, and now in the month of February also you have to prepare for the boards, practicals, and everything is there. So even from November, right, I'm not saying the complete December you have to dedicate for class 11. No, it's just that every day at least 3 hours, right, at least 3 to 4 hours you have to give. Right, every day you have to give 3 to 4 hours. What you have to do? You have to give 3 to 4 hours. Right, for what? For class, right, for what? For class 11th. Rest is for class 12th only. Rest is for class 12th only. Okay, but this is what you have to. This is the time you have to give to your class 11. See, we have wasted our 1 year. Now we have to, in this year, we have to manage both the things. And we can manage. The journey is going to be tough, but it is not impossible. Okay? It is going to be tough, but it is not impossible. So this is what you have to manage. So in December also revise your class 11, January, even till mid of the February. Then again focus on your boards because you have already completed the syllabus. And every day also you'll revise your class 12 syllabus. It's not like that you'll say complete bye to class 12 syllabus and you'll be focused on class 11th only. No, it's just that you have to revise. Okay, you have to revise. Are you getting my point? Are you getting my point? So that's what you have to understand. Then in the month of February, right, March, you'll have your board exams. You'll again revise class 12 syllabus. Okay, so read that syllabus sincerely, read that syllabus properly, so that after your board exams in the month of April, you'll have the time, right? You'll have these 15 days for your class 11th revision, right? For your class 11th complete revision. Parallelly, bache will be there for you, will take marathon sessions, will revise the thing, don't worry about that. And then give the mock test, revise your class 12th as well. So yes, you can manage it, but yeah, you have to work hard, right? You have to work hard. Okay. Okay, so are you ready for this journey? Are you ready for this journey? That yes, ma'am, we are not going to waste our time, right? We are not going to waste our time. Okay, and we will start. We will start, and this time we will not waste our class. We will not waste this year, and we will manage both the things together. So are you ready? Yes, please. Are you ready? Sure? Chali. So let's start with the basics. Okay? Let's start with the basics. So if I talk about the sexual reproduction in flowering plant chapter or the human reproduction chapter, in both the cases, we should know what mitosis is. We should know what meiosis is. These are the topics that we really need to know. Right? What mitosis is, what meiosis is, we should know. We should know, not just for the sexual reproduction in flowering plants, not just for the human reproduction, even for the genetics. We really need to know about these, right? We really need to know about these particular chapters. Okay? Like actually, we really need to know about these particular chapters. This is the first thing that you have to keep in your mind. Now, mitosis is there, meiosis is there. So, sexual reproduction, flowering plants, human reproduction, these are the things that you really need to know. Okay. So, we will start with the mitosis first. What are we going to do? We are going to start with the mitosis first. So, tell me, what do you know about the mitosis? What do you know about the mitosis? Anybody here in the chat section having any idea? Anybody here in the chat section having any idea? Yes, please. Yes, please. Yes, it is the equational division. Okay. So when you talk about the mitosis, yes, you people are right that it is the equational division. What's that? It is the equational division. What is the meaning of equational division? That parent cell will have, right? This is the parent cell. If it is having diploid chromosomes, the daughter cells will also have the same. So daughter cell and parent cell, they will have same chromosome number. This is the meaning of equational division, my dear students. This is the meaning of what? This is the meaning of equational division, that the parent cell and the daughter cell, they'll have the same chromosome number. They'll have the same chromosome number. Okay, that is the meaning of equational division. Okay, you talk about anything, right? In class 11, there is something known as growth that we study, right? There is something known as growth that we study. What is this growth? This growth is a characteristic feature. This growth is what? It is a characteristic feature. It is not a defining feature, but it is a characteristic feature. What? Growth. So how do an organism grow? We know that the cytoplasmic growth is there, plus the cell division is important, right? The cell division is important, and that is what we are discussing. That is what we are discussing. Like one of the students here in the chat section mentioning about the zygote. So if I talk about the zygote, which is a single cell, which is a single which is a single diploid cell, right? Which is a single diploid cell, and because of that zygote, you will that multicellular, right, organisms, they are going to rise from the zygote. They are going to rise from this zygote. So this zygote will undergo cleavage, right? That is also a division, right? It will become 2-celled, it will become 4-celled, 8-celled, 16, 32, so on. So cell division is something very important, right? Cell division is something very important. So even in the human reproduction chapter, right, gametogenesis is there. But after gametogenesis, whenever we will talk about this portion as well, embryonic development as well, yeah, we'll be discussing about the cleavage. We will be discussing about the divisions. So this is something we need to focus. Okay, this is something we need to focus. Are you getting my point? This is something that we need to focus. Okay, okay, done. Okay. So, as I said, mitosis, it is what? It is the equational division. What is that? Equational division. What is that? Equational division. So, what you have to understand here? Firstly, there will be karyokinesis. What is the meaning of karyokinesis? Anybody here in the chat section? We have two words here, karyokinesis and cytokinesis. Karyokinesis and cytokinesis. So, karyon. Nucleus is the word here. Kinesis, division. So, nuclear division, cytoplasmic division. Nuclear division, cytoplasmic division. These are the things that we have. Nuclear division, cytoplasmic division. These are the things that we need to study. Right? These are the things that we need to know. Nuclear division should be there. Cytoplasmic division should be there. So, when you talk about the nuclear division, there is a trick that we have, that is PMAT. What is the trick that we have here, beta? What is the trick that we have here? It is PMAT. So, P stands for prophase, M stands for metaphase, A stands for anaphase, and T stands for telophase. Are you getting my point? Are you getting my point? So, prophase, metaphase, anaphase, and telophase. Prophase, metaphase, anaphase, and telophase. Prophase, metaphase, anaphase, and telophase. Is that clear? So, this is what we have to study first in the karyokinesis. Prophase, metaphase, anaphase, telophase. So, what is the trick that we have here? It is PMAT. What is the trick, students, that we have here? It is PMAT. Everybody please repeat. What is the trick that we have here? It is PMAT. Prophase, metaphase, anaphase, and telophase. Right? This is what we have first. Now, next is what's going to happen. See, before division, before M phase, you know that we discuss about cell cycle. What we discuss? We discuss about the cell cycle. So when we say cell cycle, it is a sequential, right? It is in a proper sequence. Like right now, I'm just more focused on clearing your basics here. Okay. So if somebody is interested in completing this chapter in detail, where you discuss each and every single line, then you can follow my regular lecture as well, right? The one shots are available on this channel, right? Okay. So cell cycle, it's a proper orderly change that will take place in a cell. What is it? It is a proper orderly change that will be there in a cell. What is it? It is a proper orderly change that will be there in a cell. And what is going to happen in this particular change? Basically, the cell will grow. The cytoplasmic growth is there. The DNA replication is there. Okay, the organelles, they need to duplicate their content. So everything will happen in the cell phase. So when we cell cycle, so whenever we talk about the cell cycle, we discuss two things. We talk about the I phase and we talk about the M phase. We talk about the I phase and we talk about the M phase. So this I phase, it is interphase, it is invisible phase, it is energetic phase. Okay, so interphase, invisible phase, energetic phase, also known as I phase, also known as preparatory phase. And when you talk about this, M phase, M here is mitosis. So it is the actual division phase. What's that? It is the actual division phase. What's that? It is the actual division phase. Is that clear? Is that clear, everybody? So what's that? It is the actual division phase. Is that clear? Sure? Are you sure? Are you sure? Right? So that is what we have. So interphase, invisible phase, energetic phase, I phase, and preparatory phase. Right? This is what we have. So I phase is the word. I phase is the word. Now, here in this I phase, the cell will spend more than 95% of its lifespan in the I phase. A cell will spend more than 95% of its lifespan in the I phase. That's what you have to keep in your mind, right? It's just like your NEET preparation. You spend 2 years, 3 years so that you can give your best in this 3-hour examination, right? Right, so same is the case here. Same, genuinely same is the case here. So now when we talk about the I phase, okay, so why do, and yes, how can I forget, the most important name here is the resting phase. So why do scientists call it as the resting phase? Right, why do scientists call it as the resting phase? Is it the resting phase? Actually, no, it is not. It's just that when we observe, right, it's just that when we observe it under the microscope, when we observe the nucleus under the microscope, right beta, right, whenever we observe the nucleus under the microscope here, so we will see that cell is doing nothing. Basically, the chromatin material, it is decondensed. It is in the form of thread. Okay, so the chromosome that we discussed now, the chromosome that we form like this, the compact structure, we will only get it in the M phase. We will only get it in the M phase. That's something you have to keep in your mind. Okay, that's something you have to keep in your mind. Okay, okay, right? So this is what you need to take care. So it is the resting phase because here the chromatin material, it is decondensed, right? The chromatin material, it is decondensed. It is not condensed. It is not forming the chromosome. So that's the point that you need to take care. That's something you have to understand. Okay, that's something you have to understand. So when you talk about the I phase, you have G1 phase here, you have S phase here, and you have G2 phase here. So G1 phase is there, S phase is there, G2 phase is there. G1 phase is there, S phase is there, G2 phase is there. G1 phase is there, S phase is there, G2 phase is there. So this G1 phase, it is the gap 1 phase. It is the growth phase. It is the gap 1 phase. It is the growth phase. It is because it is after mitosis, post-mitotic phase. I think I have repeated this line that much that even I have started hating this line. There is a line in the NCERT that I phase is the phase present in between successive M phase. Right, like there is a line of NCERT that I phase is a phase which is present between successive M phase. It is the phase that is present in between successive M phase. That is what we have. Okay, so M phase, I phase, M phase. Okay, this is what we have. So when you talk about the I phase, so it is G1 phase, it is S phase, it is G2 phase. So G1, it is after M phase. So we will call it post-mitotic phase. This will be pre-mitotic phase. Are you getting my point? This will be pre-mitotic phase. Before mitosis, after mitosis. As per this line, that I phase is the phase present in between successive M phase, means present in between the M phase. So you have G1, you have S, you have G2. Right? You have G1, you have S, you have G2. So ultimately in the G1 phase, what is happening? Right? So S phase is the synthesis phase. S phase is the synthesis phase. This one is gap 2 phase. This one is growth 2 phase. And as I said, it is pre-mitotic phase. Right? And do not forget from this particular diagram as well, students, right, that this, G1 phase is the phase present in between cell division and DNA replication. That is also a very famous PYQ. Okay, it is a phase present in between It is a phase that is present in between the DNA, the cell division and DNA replication. So what do we call it? We call it as G1 phase. What do we call it? We call it as G1 phase. So that is something you need to keep in your mind. Okay, that is something you need to keep in your mind. Is that clear? Is that clear? Right? Just keep it in your mind. Now in the G1 phase, what is going to happen? What is going to happen in this G1 phase? Of course, here the RNA synthesis, the protein synthesis, all of it will be there. So yeah, the cell is metabolically very active in overall interface, right? The cell is metabolically very active. The cell is what? The cell is metabolically very active here in all these phases. And of course, see, that's a rule. This is the S phase. What's that? It is the S phase. It is the G1 phase. It is the S phase. That is the NTA's favorite question. It is the phase in which DNA replication occurs. It is the phase in which DNA replication occurs. Right? What is it? It is the phase in which DNA replication it occurs. The DNA content will get doubled. The 2C will become 4C. If the initial amount of DNA is 2C, it will become 4C. If the initial amount of DNA is 2C, it will become 4C. Right? Okay? Okay, done. Is that clear? Is that clear? So that's what you have to remember. So DNA replication will occur. So whatever is required for DNA replication in S phase, that will be synthesized here in G1 phase. Whatever is required in S phase, that will be required in that will be synthesized in G1 phase. That will be synthesized in G1 phase. So it's the gap 1 phase, growth 1 phase. So cell is metabolically very active there. So RNA will form, protein will form, all the nucleotides, ATP, whatever is required for S phase, that will be synthesized here. Most of the organelles, right? Whatever organelles the cell is having, because after that the division has to occur. Okay, the division has to occur. The parent cell will form 2 daughter cells, right? The parent cell will form 2 daughter cells. Are you getting my point? The parent cell will form 2 daughter cells here. And what is going to happen? See, these parent cells, they need to distribute their DNA content equally to these daughter cells. Along with that, even the organelles, right? They need to. Yes, bache, they need to. Right? They need to distribute the organelles. Right? They need to distribute the organelles as well. Done? Done? Okay, right? So G1 phase, that is going to happen in the G1 phase. And overall, the G1 phase, its duration is variable. Its duration is what? Its duration is variable. Its duration is what? Its duration is variable. Are you getting my point? What will happen to its duration? Its duration is variable. Am I clear here? Am I clear here? Yes? Am I clear here? Okay, so that's something you need to keep in your mind. Okay, that's something you need to keep in your mind. Okay? Done. Now in the S phase, as I said, the DNA replication will occur, right? If the initial amount of DNA is 2C, it will become 4C, right? If the initial amount of DNA, if it is 2C, it will become 4C. That's what you have to keep in your mind. This is what your S phase is. Are you getting my point? This is what your S phase is. Is that clear? This is what your S phase is. Is that clear? Is that clear? Keep it in your mind. Now, there are another things that we need to know here, right? See, S phase. Again, I'm repeating. So, what is the word, people? What is the word here? It is the S phase, the synthesis phase. What is the word here, people? It is the S phase. It is the synthesis phase. So, as I said, DNA amount will get doubled. DNA amount will get doubled. What will happen here? DNA amount will get doubled. That's what you need to keep in your mind, that it is the DNA amount that will get doubled, number one. So that is why in the NCERT it is written that if the initial amount is 2C, it will become 4C. It will become 4C. So if the initial amount is 2C, it will become 4C. But number of chromosome will remain same. Number of chromosome will remain same. Okay, number of chromosome will remain same. That's something we need to keep in our mind. That's something we need to keep in our mind. Okay, okay. So DNA amount will get doubled. If initial amount is 2C, it will become 4C, but the number of chromosome will remain same. Okay, but the number of chromosome will remain same. Now let me give you an example. You have a cell. Let's suppose 2n is equals to 4. 2n is equals to 4 means, what is the meaning of 2n is equals to 4? 2n is equals to 4 means the diploid number is 4, right? The diploid number is 4. Is that clear? Is that clear? The diploid number is 4, right? That is the meaning of 2n is equals to 4. Now, normally, this type of chromosome, be it this or this, you will see, right? You will see it only, right? You will see it only when the cell is dividing. That is in my prophase. That is in prophase. That is when? That is in prophase. Done? 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 So now see, this is also a chromosome. This is also a chromosome, right? This is also a chromosome. This is also a chromosome. They both are chromosome. Now in this chromosome, what's that? It is centromere. What's that? It is centromere. And this is also a chromosome, my dear students, right? It's not like that this is chromosome and this is not. They both are, right? But the difference is, I'll tell you the difference. And here, you'll call these arms as sister chromatids because they are attached to the same centromere. They are attached to the same centromere. Is that clear? Is that clear? Is that clear? Now I'm giving you an example. As I said, you'll see the chromosome like this when the chromatin will be condensed, right? When the chromatin will be condensed, this is what you people are going to see. Are you getting my point? This is what you people are going to see. Are you getting my point? Right, people? So, because I have to give you the example, that's why I'm telling you it in this way. Let's suppose there is a cell having 4, the chromosome number. So, right, before this S phase, that is how it is going to be. Total, how many chromosomes are there? 4. Now, when you look at it, it is monad. Monad means this chromosome is having single arm. And as I said, the compacted structure, you will see in prophase, but I'm giving you the example here. No doubt in interphase, it will be like this, in the form of a thread, right? And it is having single arm. So, we will call it as monad, right? We will call it as monad. We will call it as monad. Are you getting my point? Are you getting my point? Now, what's the point here? After S phase, right? Now here, initial amount of DNA will be 2C. Matlab C is basically just showing that, amount, the quantity, nothing else, right? C is just telling us this is the amount, this is the quantity, that's it, right? 2C, initial amount it is. What is it? Initial amount it is. What is it? Initial amount it is. So it is the 2C, okay? So you can say 100 picogram or something, whatever quantity you want to take. After S phase, see, ultimately this is also DNA and protein. So after S phase, the DNA content will get doubled, right? This monad will become dyad. This monad will become dyad. What is the meaning of that? right? What is the meaning of that? The monad will become dyad. So basically that is how it is going to be. That is how it is going to be. Right? That is how it is going to be. 1, 2, 3, 4. Here also it is 1, 2, 3, 4. Okay? It's just that the DNA amount got doubled. It will become 4C in S phase. It will become 4C in S phase. The DNA amount will get doubled. Okay? The DNA amount will get doubled here. Right? So 1, 2, 3, 4, 1, 2, 3, 4, it will become 4C. That's what you need to keep in your mind. So 2C will become 4C. 2C will become 4C. 2C will become 4C, but number of chromosome is same. That's what you have to understand. And even in the S phase, the centriole duplicates. Centriole, you know na, non-membrane bound organelle. It is the centriole that duplicate. It is the non-membrane bound organelle. It is a non-membrane bound organelle. So that's something you have to keep in your mind. Again, in the G2 phase, you know, the same thing. The RNA synthesis, the protein synthesis, right? It will occur. RNA synthesis, protein synthesis, that will occur. That's what you have to keep in your mind. Okay, that's what you have to keep in your mind. So RNA, protein, they all will synthesize, right? They all will synthesize. And here in the G2 phase, the tubulin synthesis is also there. What will be there? Tubulin synthesis will be there. Okay, tubulin synthesis will be there. Is that clear? Is that clear? Sure? Are you sure? Any doubt? So this is about the, right? This is about the interphase. Now, we have to talk about the prophase. What we have to discuss, people? We have to discuss about the prophase. So, whenever you talk about the prophase, let's suppose this is your cell, this is the nucleus here. Here you will have this structure. We call this structure as centrosome. What is the meaning of centrosome? See, centrosome is having another name as well. You can call it cell center. You can call it diplosome. Centrosome, cell center, diplosome, and sometimes cell sphere as well. Sometimes cell sphere as well. Okay, so centriole or centrosome, you will see it in the animal cell. Where will you see it? You will see it in the animal cell, not in the, right? You will see it in the animal cell, but not in the plant cell. Not in the plant cell. Keep it in your mind. Chalo. Now, when two centrioles perpendicular to each other, when they are covered with, right? When they are covered with what? Pericentriolar material. When two centrioles perpendicular to each other, when they are covered with pericentriolar material, which is the amorphous substance, then we call it as centrosome. And here in prophase, you will see that slowly nuclear membrane will disintegrate these centrioles, centrosomes, because what will they do? This pericentriolar material will do the polymerization of the tubulin protein. It will form the microtubules, right? So basically that microtubules will try to push each other. So basically overall, right, in a very quick way we are revising, these centrioles, right, centrosomes rather, they'll move to the opposite pole. So by the end of the prophase, right, because the fiber synthesis is also there, that fiber will push each other and finally they'll reach to the opposite pole, right? Plus your nuclear membrane also disintegrate, right? Your nucleolus disappear, all the organelle content, it will disappear. Okay, it will disappear. Your chromatin material, it is condensing, it is losing water, it is becoming compact, and by the end it will get the shape like this, right? The shape like this. So by the end of the prophase, so basically prophase will mark the initiation, the beginning, the initiation of the compaction, initiation of condensation of chromatin material. Initiation of condensation of chromatin material will be there. What will be there? Initiation of chromatin material or the genetic material will be there. So chromosomes will form like this, right? Chromosomes will form like this. Okay, and as I said, the pericentriolar material here is for the nucleation. It will form these fibers, right, bache? It will form these fibers. So yeah, by the end it will be something like that. The genetic material is there in the cytoplasm, the fibers will attach to it. But how will they attach, right? So see, this is your chromosome, this is the centromere, right? So it is covered by the disc-shaped proteins. What do we call it? We call it as kinetochores, right? It is covered with the disc-shaped protein. We will call it as kinetochore. What will we call it? We'll call it as kinetochore. So these fibers will attach to it like this, right? So this is a PYQ that these chromosome fiber, they never get attached to the centromere directly. They will always attach to kinetochore. They will always attach to kinetochore. So by the end, like these chromosomes, they'll be scattered here in the cytoplasm, right? And these fibers, they are all attached. So whenever you talk about the spindle fiber, you will see, because in animal cell centriole is there. So these astral rays they form. We use the word aster for it. Aster means star-like, right? Some fibers, they'll come out like this. They'll come out like this. And even other than that, there will be some continuous fibers, there will be some discontinuous fibers. So what they have to do, ultimately they have to form the spindle apparatus, right? So prophase is marked by, right, the initiation of chromatin material will be there, the condensation of chromatin material will be there. That is something you have to keep in your mind. Okay, that is something you have to keep in your mind. Is that clear? Is that clear? In the prophase, that's going to happen, right? So spindle fibers, spindle apparatus will form, nuclear membrane will disintegrate, nucleolus will disappear, right, bache? The centrioles, they will move to the opposite pole. Okay, all the organelle content, it will get duplicated, it will get doubled, it will get disappear. It will be doubled in the interphase only, it will be disappear. Now in the metaphase, very simple it is, right? By the end of the prophase, they are just scattered. Right, they are just scattered there in the cytoplasm. Now they'll arrange itself at equator. They'll arrange itself at equator. They'll arrange itself at equator. Like this. Like this. Okay. So it is the metaphase in which the morphology, it is the metaphase in which we can study the morphology of the chromosome, in which cell shape, size, number will be studied. Okay, cell shape, size, number will be studied. Okay. Okay, so always remember these fibers, they are attached to the kinetochore, the disc-shaped protein that are covering the centromere, but not directly. Then after that, there will be anaphase. So from this diagram itself, I will make it clear for all of you. So in the anaphase, what's going to happen? There will be the splitting. Right, there will be the splitting, because we have to divide the equal number of chromosome to both the daughter cells. We have to divide the equal amount of genetic material as well. So in anaphase, right, what's going to happen? There is the splitting of centromere. There is the splitting of centromere and separation of sister chromatid. Splitting of centromere and separation of sister chromatid. Okay, that's going to happen. And finally, students, in the telophase, what will be there? In the telophase, what will be there? It is the reversal of prophase. So whatever happened in the prophase, that will be reverse. Like nuclear membrane will again form, because now the chromosomes, they are at the opposite pole. Now see, when there is a separation, when there is a separation like this, so now these sister chromatids, they'll start behaving like the chromosome. These sister chromatids, they will start behaving like what? They will start behaving like the chromosome. They'll be the chromosome. They'll reach to the opposite pole. They will decondense here. They'll form loose threads there. Again, they'll lose their identity as the discrete units. The nuclear membrane will again form, organelles will again appear. That's why in simple words we say that it is the reversal of prophase. In simple words we say it is the reversal of prophase. Then cytokinesis is there. So in animal cell, cleavage furrow will form, and in plant cell plate. That's it. Now meiosis, which is the reductional division. Now meiosis, which is the reductional division, which is what? It is the reductional division. What is it? What is this meiosis, students? This meiosis is the reductional division. This meiosis is what? This meiosis is the reductional division. It will reduce the chromosome number to half. So that's how it occurs. That's how it is. You pay attention, you'll get to know about it. Okay. You just pay attention, you'll get to know about it. So what do we have? Right, it is the meiosis, reductional division. So firstly, meiosis 1 will occur. Firstly, meiosis 1 will occur. Then there will be meiosis 2. Meiosis 1 will occur and then there will be meiosis 2. Meiosis 1 will occur and then there will be meiosis 2. Are you getting my point? So meiosis 1, you will get 2 cells, 2 haploid cells, right? 2 haploid cells. Remember, the DNA amount is 4C here because it is after interphase. Right, here it will be 2C and 2C, then there is meiosis 2, so the DNA amount will be this. So if you will see, if you will look at it, there will be 2 rounds of, right, there will be 2 rounds of nuclear division and cytoplasmic division. There will be 2 rounds of nuclear division and cytoplasmic division. There will be 2 rounds of nuclear division and cytoplasmic division and 1 round of DNA replication. 1 round of DNA replication. Okay, that is going to happen here. 2 rounds of, what will be there? There will be 2 rounds of, right, nuclear division and cytoplasmic division, and there will be 1 round of DNA replication. Okay, matlab that will occur just once. Done? Okay. Okay. Any doubt? Any doubt? Right? So that is going to happen. So overall here again in meiosis 1 you will see prophase 1, metaphase 1, anaphase 1, telophase 1. So prophase 1 will have 5 substages. That is your leptotene is there, also known as leptonema. Zygotene is there, also known as zygonema. Packeting, in which crossing over occurs, is there, known as pachynemia. Then there will be diplotene, and finally there is the dikinesis. Okay, dikinesis. After the telophase 1 people, there will be prophase 2, there will be metaphase 2, there will be anaphase 2, there will be telophase 2. So in between telophase 1 and prophase 2, that is interkinesis. That is interkinesis. Is that clear? Is that clear? So that's something you need to keep in your mind. That's something you need to keep in your mind. Is that clear? Is that clear? So yes, this is something you really need to understand right before starting your class 12 syllabus. Now, the next thing is little bit from biomolecules. Okay, you remember when we study the biomolecules, we say bio macromolecules are there, we say bio micromolecules are there. In macromolecules, molecular weight is above 10,000 Dalton. Is above 10,000 Dalton. Here it is less than 1,000 Dalton, right? Less than 1,000 Dalton, right? Right? This is what we know. Okay? Done? So here we basically talk about the polymers, right? So for an example, you have proteins, polysaccharides, nucleic acids. They all are what? They all are biomacromolecules. They all are biomacromolecules. What are they, people? They are biomacromolecules. Okay, biomacromolecules. Okay, number 1. Number 2, right? Number 2, this is number 1. Protein is there, polysaccharide is there, nucleic acid is there, right? They all are what? Biomacromolecule. Now, nucleic acid. When we talk about the nucleic acid, we discuss DNA and RNA. And trust me, we need them a lot in our genetics, right? We need them a lot in our genetics, right? DNA and RNA. So it's deoxyribonucleic acid. It is ribonucleic acid. It is ribonucleic acid, right? It is deoxyribonucleic acid. It is ribonucleic acid. That is what we have. It is deoxyribonucleic acid. It is ribonucleic acid. So when you are talking about the nucleic acid, they are the polymer, or you can say that they are the polynucleotide chains. What are they? They are the polynucleotide chains. What do we call them, people? We call them as polynucleotide chains. Is that clear? Is that clear? Is that clear? So molecular weight is this, right? Molecular weight is this, right? So DNA is there, RNA is there. DNA is there, RNA is there, right? So deoxyribonucleic acid, ribonucleic acid. Deoxyribonucleic acid, ribonucleic acid. So they are ultimately the polynucleotide chain. So we need to understand that what is the meaning of nucleotide here. We need to understand that what is the meaning of nucleotide here. So to understand nucleotide, right, it is made up of nitrogenous base, sugar plus phosphate. Okay, now firstly we need to understand what nitrogenous bases are, and they are biomicromolecule. They all will be together to form biomacromolecule. So the first word that we have here is, right, the first word that we have here is the nitrogenous base. Right, people? What is the first thing, first word that we have here? It is the nitrogenous base. It is the nitrogenous base. What is the word that we have here? It is the nitrogenous base. Are you getting my point? So when we discuss this thing, this is nitrogenous base. Here, you need to know about two things. Purines, pyrimidines. Puga and cut. That's the trick that we have. So when it is puga, right, pu means purines, G means guanine, A means adenine, A means adenine. So from this 9 and 9, you have to remember 9-membered ring. You have to remember 9-membered ring. It's a 9-membered ring. It's a 9-membered ring. What's that? It's a 9-membered ring. And here in adenine and guanine, as I said, in adenine and guanine, what's that? It's a 9-membered ring. What's that? It's a 9-membered ring. What's that, people? It is the 9-membered ring. Okay, so here in this 9-membered ring at 1, 3, 7 and 9, nitrogen is present in ring. Take nitrogen is present in this ring. Do revise it in detail, right? Next, pyrimidines. So when you people talk about the pyrimidines, I will not show you the structures today, okay? So that's what you have to do. That will be your homework. So it will be cytosine, it will be uracil, and it will be thymine. This thymine is also known as 5-methyl uracil. It is also known as 5-methyl uracil, and it is a 6-membered ring. What's that? It is a 6-membered ring. What's that? It is a 6-membered ring, right? So cytosine is there, uracil is there, thymine is there. Now, sugar. Sugar, what are they? They are the monosaccharides. What are they, people? They are the monosaccharides. What are they, people? They are the monosaccharides. So mono means a single. Mono means single. Saccharide means sugar. Mono means a single. Saccharide means sugar, right? It's a single sugar. It's a pentose sugar. Why pentose? Because the carbon number is 5 here. Why pentose? Because the carbon number is 5 here. So when you talk about the sugar, we have the ribose, we have the deoxyribose. So these sugars, from the name itself, that's what you have to remember, that RNA will contain ribose sugar, DNA will contain deoxyribose sugar. DNA will contain deoxyribose sugar. DNA will contain deoxyribose sugar. Clear? Clear? Yes, is that clear? Sure? Done? So RNA is there and DNA is there. RNA is there and DNA is there. Take, this is what we have. All right. Now, if I'll show you the structure, it's something like that. This is the first carbon, HOH, 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 H, CH2OH. HOH, H, H, HOH, H, CH2OH. So it is deoxyribose. What's that? It is deoxyribose. Here at second carbon, here at second carbon, where at second carbon O is missing, right? O is missing. That's what you have to remember. And in RNA, my dear students, in RNA you will see cytosine and uracil is there, adenine and guanine is there. In the case of DNA, cytosine and thymine, adenine and guanine. So uracil is there in RNA only, it is not there in the thymine. Understood? Yes, please. Is that clear? Done. So general formula for monosaccharides is CnH2nOn. The general formula for monosaccharide is CnH2nOn. It is CnH2nOn. So C is 5 here, so it will be C5H10O5 and deoxyribose is C5H10O4. So beta, it is not, it doesn't even follow the general formula. It doesn't even follow the general formula. Is that clear? It doesn't even follow the general formula. Okay? Okay? Sure? Are you sure? Are you sure? Okay? Chali. So, next is beta, in the nucleic acid you will see phosphoric acid is there. What is there? Phosphoric acid is there. What is there? It is H3PO4. What's that? It is H3PO4. So it is the phosphoric acid that is present. What is present? It is the phosphoric acid that is present. Phosphoric H3PO4. Like that is how it is now. Then that is what we have. So basically what's going to happen? Your nitrogenous base, when it joins with sugar, N plus S, it will give you nucleoside. Right, so N stands for nitrogenous base here, S stands for sugar. So when your nitrogenous base plus sugar plus it is the phosphate, it is known as nucleotide. It is known as nucleotide. Is that clear? So tide, T means 3. There are 3 things that will join. That's how you can remember. So nitrogenous base plus sugar, that will be N glycosidic bond that is going to connect them. So here also in nucleotide, Nitrogenous base plus sugar, they bind with N-glycosidic bond, my dear people, and sugar and phosphate, they have phosphoester bond. They have phosphoester bond, right? They have phosphoester bond. Take. So adenosine, that's a nitrogenous base. Guanosine, that's a nitrogenous base. Cytidine, that's a nitrogenous base. Uridine, that's a nitrogenous, sorry, that's not a nitrogenous base. They all are nucleoside. They all are nucleoside. Take. So adenine is nitrogenous base, guanine is nitrogenous base, cytosine is nitrogenous base, uracil is nitrogenous base. When it is adenosine, guanosine, cytidine, uridine, this is the nucleoside. Same way, when it is deoxyadenosine, deoxyguanosine, deoxycytidine, or deoxythymidine, because in DNA we do not have uracil and it is also known as thymidine. So they are what? They are the nucleoside. Nucleoside. Now in the nucleotide, one phosphate has been added. So you can call it like this: adenosine monophosphate. So the nomenclature is important, right? Guanosine monophosphate, cytidine monophosphate, uridine monophosphate. Okay, so the another names here are adenylic acid. So adenosine monophosphate, adenylic acid is same. Guanylic acid and guanosine monophosphate is same. Cytidilic acid and it's same. Uridylic acid and it's same, right? So this is about the nucleotide. So yes, before starting the molecular basis of inheritance, you should get an idea of this. You should get an idea of this. Understood? Understood? So rest is like this. Sugar is sweet. Everybody is interested in having bond with sugar. Phosphate. Sugar, nitrogenous base, phosphate. Sugar, nitrogenous base, phosphate. Take. So they also join with each other. They also join with each other. So it is the phosphodiester bond that is going to connect it, join it. So this is the basics here. Okay, and this is something you really need to, right? Genuinely, this is something you really need to know, right? You really need to know if you are interested in starting your class 12th at a very, very, very good level. So look at this DNA part, 2 nanometer long. So ultimately, what's that? Sorry, 2 nanometer diameter, 20 angstrom is the diameter. Okay, so it's ultimately the polynucleotide chain where the nitrogenous base is there in the center. Adenine and thymine will have 2 hydrogen bonds. Cytosine and guanine will have 3 hydrogen bonds. Is that clear? Is that clear? So 2 hydrogen bonds, 3 hydrogen bonds. That is what we need to remember here. Okay? Okay, any doubt? Yes, please. Any doubt? Are you sure? Are you sure? So these are the basic things that you need to understand, okay? That you need to understand. Now, if you guys are having any doubt, firstly, let's discuss that and then let's start the next part. Yes, then we will start the next part. No doubt. Madam, we are not even listening. So how can we get the doubt? Chali. So students, do not forget that till 31st of March, okay, till 31st of March, right, you people can avail our plus subscription, right, just at rupees. Again, it is not working. Again, it is not working. right? But don't worry, after a minute or two, it will be replaced. This link will be replaced. So you can just click on this particular link and you can join, right? You can join the Nexus batch. You can join R Plus subscription, right? But that too at just 4499. So if you ask me the team that who are going to teach you, so you know that you can see the faces here. So we are about to start the marathons. Okay, so your HSP sir, right? Baba HSP. And he's having the, I think, yeah, he's teaching from last 27 or 28 years, right? Last 27 or 28 years. He's that experienced. When it comes to chemistry, nobody can beat him. I can, like, you come up with anybody, right? I have so much faith in him now, right? And it's not just me. You can just ask the same thing from his students as well. He's amazing when it comes to chemistry, right? He's amazing. And you already know about the Wassim sir. Fine, you already know about the Wassim sir. So Wassim sir and HSP sir, they both are going to teach you the chemistry. And trust me when I'm saying, when we talk about the HSP sir, even Wassim sir, we all appreciate him a lot. So he is having the teaching experience of more than 27 years. And when it comes to chemistry, nobody can beat him, right? You pick up anything, organic, inorganic, physical. He's the guru. He's the Baba. Right, then I'll be teaching you the biology, right? I will make sure that you understand each and everything from the NCERT. I'll make sure that you'll be able to decode your NCERT, okay? And your Yava sir will be teaching you the physics. So this is the amazing team, right? That is going to mentor you, that is going to guide you, okay, people? So without any delay, be the part of our batches. And for NEET 2025 aspirants, you know that yesterday we have announced the 15 mega marathon sessions where we are going to complete the syllabus. So I'll be starting with this, right? I'll be starting with this complete class 11th zoology. And yes, here we are going to start with the animal kingdom, right? Then of course we will pick up the cell cycle and cell division, not exactly cell cycle and cell division. Then we'll jump to the structural organization and then we'll talk about the human physiology. So that is what we are going to complete in this particular marathon. So if you are going to attend it, definitely you'll complete the most important topics of zoology of class 11. So come on, hit the like button, everybody. Hit the like button quickly. Mark your attendance if you are appearing in the NEET 2025. And if you are in class 12th, right, and you are looking for a session where you can revise your entire class 11, these sessions are for you. These sessions are literally for you, people. Okay, right? These sessions are literally for you. So what you have to do, click on this particular session, right? Hit the like button here, and here also, beta, you have the links, right? You have the links in the description. You can click there, and you can be the part of our batches. Okay. Okay. So this is all about today's lecture. So whatever doubt you have, right, do let me know in the comment section. If you like the session, do let me know in the comment section. So take care, everybody. Thank you so much. See you all soon.